The best way to understand the genetic differences that we find in human populations is that populations differ by distance. And it's a continuous change from one group to another. And one way we can look at this is use the example of skin color. If we were to only look at people in the tropics and people in Norway, we'd come to the conclusion that there's a group of people who have light skin, there's a group of people who have dark skin. But if we were to walk from the tropics to Norway, what we would see is a continuous change in skin tone. And at no point along that trip would we be able to say, oh, this is the place in which we go from the dark race to the light race.
By the 1920s, a single drop of blood reflecting African ancestry could identify any individual as black and inferior in every way. In the not-so-distant past, many of these students would have been considered contaminants were they to have bred into the superior white race. 28 states passed laws forbidding intermarriage to safeguard white racial purity. Racial purification was one aim of the eugenics movement. The science of eugenics rested on simple Mendelian genetics. One gene each from father and mother, it was believed, gave rise to any trait, physical, behavioral, even moral.
Some of these things were things like the ability to play chess, rowdiness, congenital feeble-mindedness, virtually any cultural or behavioral trait you could imagine. Now, the mistake that they were making was assuming that complex behaviors could be reduced to simple Mendelian genes.
Nonetheless, eugenicists used the science of the day to advance a social agenda widely accepted in white America. To breed the best and the brightest, always white, and breed out society's worst and weakest, of all colors.
There's a lot of concern about race mixing. You don't want a superior race, a race with great qualities of intellect and achievement and musical genius and these kinds of things to mix with a race on a lower stage of civilization that has fewer of these characteristics, because that again would bring down the level of those characteristics and what you want to have for your civilization.
If human variation were to map along racial lines, people in one so-called race would be more similar to each other than to those in another so-called race. That's not what the students found in their mtDNA. What about other genetic differences?
The problem for evolutionists and population geneticists was always to try to actually characterize how much genetic variation there was between individuals and groups. And I spent a lot of time worrying about that, like other people in my profession.
In the 1960s, Richard Lewington decided to find out just how much genetic variation fell within and how much between the groups we regard as races. A new technology enabled him to do pioneering work.
And that method, which is called gel electrophoresis, a very fancy name, we were able to use on any organism at all. If you could grind it up, you could do it. That included people. I mean, you have to grind the whole person, but you could take a little bit of tissue or blood. Over the years, a lot of data were gathered by anthropologists and geneticists looking at blood group genes and protein genes and other kinds of genes from all over the world. I mean, anthropologists just went around taking blood out of everybody. I must say, if I were a South American Indian, I wouldn't have let them take my blood, but they did. And so I thought, well, we've got enough of these data. Let's see what it tells us about the differences between human groups.
Lewington's findings were a milestone in the study of race and biology.
And if you put it all together, and we've now got that for proteins, for blood groups, and now With DNA sequencing, we have it for DNA sequence differences. It always comes out the same. 85% of all the variation among human beings is between any two individuals within any local population, between individuals within Sweden or within the Chinese or the Kikuyu or the Icelanders.
To put it another way, of the small amount of variation in our genes, there is apt to be as much difference between Gorgeous and her teammate Christine as there is between Gorgeous and her opponent Kaylin. Any two individuals within any so-called race may be as different from each other as they are from any individual in another so-called race.
Are the people who we call black more like each other than they are like people who we call white, genetically speaking? The answer is no. There's as much or more diversity and genetic difference within any racial group as there is between people of different racial groups.
Still, we know that some genes are found with greater frequency in some populations.
And geography is the better way to explain that more than race or anything else. There can be accumulations of genes in one place in the globe and not another.
Like the gene forms regulating skin color. And for some genetic diseases, like sickle cell disease. Long assumed to be a racial trait, sickle cell disease is a debilitating disorder caused by a gene form that alters the shape of red blood cells.
It's one of the misconceptions that sickle cell disease is an African American or an African disease. The sickle cell trait is not uncommon in people from the Mediterranean region. In fact, in some parts of Greece, up to 30% of people in the population may carry sickle cell trait. Sickle cell trait persists in certain populations around the world because of the relative resistance it confers to malaria. So people who've got sickle cell trait are less likely to develop malaria, and when they do develop it, they are less likely to develop severe complications and to die from it.
And so understanding how evolution works really helps to understand this a lot. While nobody would want sickle cell anemia, in areas that have malaria, if you have sickle cell trait, if only one of your parents passes on the sickle cell gene to you, then you can have sickle cell. It doesn't kill you, but it will inhibit malaria from killing you. And so the natural selection of evolution then will select for that, not like an intellectual an intellectual selection, but because they survive more in that area. And so through Africa, through areas of the Mediterranean, and through India are all areas of high malaria. So those in those regions that had the sickle cell trait, that only got it from one parent, were able to live, pass on their genes because they could have kids because they lived long enough, and so then it becomes part of those populations. A large part of that is Africa, and since African Americans were the largest part of the population here, and we had the race idea, then Africans, the black race, have this problem. But in fact, much like the idea of skin color, it is environmental. And as you look at evolution and how things are selected, it's cause and effect, processes and procedures, and pressures.
Where malaria was common, the sickling gene was selected. In Arabia, South Asia, Central and Western, but not Southern Africa. And in the Mediterranean basin, the home of Jackie Washburn's ancestors.
So I don't think we're ever going to be able to isolate a gene for athletic performance.
Or a gene for any complex trait. If genes contribute to Marcus's musical talent, There would be dozens, interacting with environment, training, and practice. Those genes would be inherited independently of the genes for eye shape, skin color, and hair form, which Marcus inherited through his Korean and Jamaican ancestors.
For race to be more than skin deep, one has to have concordance. In other words, skin color needs to reflect things that are deeper in the body under the skin. But most of human variation is non-concordant. Skin color or eye color or hair color is not correlated with height or weight. And they're definitely not correlated with more complex traits like intelligence or athletic performance.